Also, their interaction as a part of broader ecological system has to be recognized, uh, in that they interact with and affect other parts of systems such as the adjacent water bodies or the groundwater. So all of these functions, all of these variables have to be considered when defining soil health. However, in an agricultural context, a simple definition of soil health is that it's the fitness of soil to be able to support crop growth without becoming degraded or otherwise harming the environment. So what happens when the soils are unhealthy? The slides here shows an unhealthy soil towards the left of the screen and a healthy soil towards the right. Unhealthy soil seems to lack any aggregation. It has wide cracks. The grayish color indicates not much organic matter in it. The healthy soils on the right of the screen have large aggregates. They are rich in organic matter. The water infiltration and retention in such soils is generally high, so not much runoff is produced in these kind of soils. So the potential problems that arise in an unhealthy soil is a lack of soil structure due to lack of aggregation, which also leads to crusting of the soil or the compaction, especially if heavy farm operations are carried out on these soils. Irrigation and rainfall water is lost due to excess runoff, and also that causes a deficit of moisture in these soils, which ultimately leads to the loss of biodiversity. All of these problems are linked to different soil properties, different physical, chemical, and biological properties of the soil. For example, lack of soil aggregation leads to poor soil structure and compaction. Less organic matter leads to poor nutrient cycling and water retention. Similarly, water infiltration is linked to erosion. So these different physical, chemical, and biological properties interact amongst themselves to provide a healthy soil. And these properties can be used as parameters of soil health measurement. It's also important to note that these are only that using a subset of these properties will not be sufficient to manage soil health. And a comprehensive assessment of soil health should include the physical and the biological components, along with the chemical properties of soil that are generally evaluated in the soil testing labs. So I'll discuss some of these soil health parameters in the presentation. I will focus more on the biological and the physical components, since the chemical components are generally quite well known, since they form the basis of the chemical fertility recommendations of the soil testing labs. A healthy soil consists of several species of mice, gophers, insects, earthworms, nematodes, and also microorganisms, including hundreds of species of fungi, thousands of species of bacteria. This huge diversity of organisms is generally not evenly distributed in the soils, since there are localized microclimate zones in the soils containing areas of good and poor aeration, cool and warm temperatures, moist and dry conditions. However, this diversity is useful. It is an indicator of good soil health. It leads to the diversity in function such that the soil organisms are able to utilize a wide variety of substrates that are available in soil to carry out a wide variety of processes in the soil. The species diversity also ensures that if environmental conditions change, the soil processes can still be carried out, since other different species are capable of carrying out those tasks under those different conditions. So it also provides stability and resilience to the soil. One of the most important organisms in soil is the earthworms. They eat their way through the soils and form a a lot of burrows. An earthworm can ingest 2 to 30 times its own weight of soil in a single day. And a population of earthworms can ingest anywhere between 50 to 1,000 tons of soil per hectare in creating an extensive system of burrows. These burrows act as macropods. They break up large soil clods, and they also offer paths of least resistance to the growth of plant roots. The ingested soil is expelled as casts or globular soil aggregates. These casts have higher concentration of polysaccharides, which are sticky substances, and they help in enhancing the soil aggregation. The casts are also rich in bacteria from earthworms' gut, and the nutrients in these casts are also broken down into plant available forms due to the activity of digestive enzymes in the earthworm. So the activities of earthworms not only alter the soil physically through burrows, they also alter it chemically through the casts, and in this way they are responsible for increasing the productivity of soils. This infographic here from the USDA shows the richness of soil or microorganisms in soils. A full of soils contains about 100 million to a billion different bacteria. Fungi are a little bit less, accounting for about a million. But due to the larger size of fungi, they actually have a higher biomass and higher metabolic activity compared to the bacteria. I'll be using this graphic of nitrogen cycling to show the major functions of this huge population of microorganisms in the soil. So the most important contribution of microorganisms is the decomposition of plant residues. Dead leaves, roots, and other plant parts are broken down so that the nutrients in these parts are released back into the soils for their uptake by the plants. Fungi play an especially important role in this decomposition, especially of complex organic materials, which cannot be further decomposed by other microorganisms like the bacteria or the actinomycetes. The mineral forms of these elements, including nitrogen, phosphorus, sulfur, etc., are released into the soil solution on the decomposition of organic residues. Many of the enzyme-mediated transformations of these minerals are then carried out, especially by bacteria, since they are responsible for a wide array of enzymatic activity. For example, specific nitrifying bacteria are required to carry out the nitrification process to convert the ammonium into plant available nitrates. One of the other important functions for bacteria is the nitrogen fixation. The bacteria convert atmospheric nitrogen to plant available nitrogen compounds. In agricultural soils, generally the rhizobium bacteria perform this function in association with the root nodules of the legumes. And they are responsible for enormous quantities of nitrogen fixed into plant usable forms throughout the world. One of the similar activities of fungi is their beneficial association with the higher plant roots. This association is called mycorrhizae, which means fungus roots. In this symbiotic association, the fungi obtain their sugars directly from the plant roots, and in return, the fungi contribute to the extension of the plant root system by 5 to 15 centimeters. Also increase their absorptive surface area, which helps in the uptake of soil nutrients, especially the immobile nutrients like phosphorus. They can also help with the uptake of water, thus helping with the drought resistance for plants, and also sometimes protect them from soil-borne disease or fungal pathogens by competing for the infection sites. The fungal hyphae also play an important role in promoting soil aggregation along with the plant roots. The slide on the left part of the screen here shows white fungal hyphae that help in binding the soil particles and tiny microaggregates into larger macroaggregates. The hyphae and the plant roots also exude sugar-like substances which are sticky in nature and act as cementing agents. Similarly, the mycorrhizae also secrete a sticky proteinaceous substance called glomalin that is very effective as a cementing agent. We've already discussed the role of earthworms in promoting soil aggregation through the formation of burrows and casts. Similarly, the microorganisms like bacteria produce sticky organic substances that also help in soil aggregation. All of these different biological agents play an important role in the formation of especially the bigger soil aggregates called the macroaggregates from the microaggregates. This graphic here shows macroaggregates with a network of pore spaces in between them. The aggregation of soil determines the amount and the interconnectivity of these pores. Large aggregates lead to lower bulk density and a higher proportion of macropores, which aid in optimal movement of water, nutrients, oxygen, and heat to the plant roots. They also help in supporting the plants and also in the penetration of roots. A proper structure also provides proper habitat for soil microbes and flora and fauna of soil. One of the other important properties of aggregates is their stability or their ability to resist the laws of structure due to external forces like rainfall, irrigation, or tillage. There are the following videos. These videos here show the stability of aggregates for different kinds of soils. This is from topsoil sample. These soils have nicely formed aggregates. You can also see some of the roots, and they hold up nicely when we apply water to them, indicating that aggregates are held up pretty strongly. When we move down the horizon to the sea horizon, these soils have lower organic matter content, not so many roots. Aggregation is lesser, so these soils are disintegrating much quickly compared to the topsoils, thus showing the importance of roots and organic matter in providing stability to these aggregates. The topsoils with cover, with a cover crop, lots of roots here, lots of organic matter. These soils show almost no disintegration. The water remains almost clear, again indicating the importance of roots and organic matter in holding up the aggregates. This topsoil does not have any cover. The aggregation is, there is not so much aggregation, and it sort of disintegrates quickly when the water is applied compared to the soils with a cover crop. There's quick disintegration of aggregates here. Similarly, in highly saline soils, especially when there is high content of sodium, uh, can lead to dispersion of soil particles and disintegration of soil aggregates very quickly. As shown here, the aggregates are disintegrated very quickly on the application of water. So what happens with the disintegration of aggregates is that it leads to the destruction of macropores. The pore network here, which is responsible for the infiltration of rainwater or irrigation water. In the absence of proper infiltration, water runs off the surface, thus removing the topsoil layer and leading to water erosion and moisture deficiency in these soils. One of the most important parameters for soil health is soil organic matter. It exerts a dominating influence on many soil physical, chemical, biological properties. The previous videos showed the importance of organic matter in maintaining aggregate stabilization. Its cementing properties holds silt and clay particles together, thus forming as well as maintaining the soil structure, and it reduces losses via the wind and water erosion. It's a source of energy and nutrients for soil microorganisms. Thus the biological component of soil is dependent on it. Soil nutrients such as nitrogen in organic matter is released gradually through the decomposition of its mineralizable fractions, and thus it's a nutrient storehouse, making the nutrients available slowly for uptake by plants. It also accounts for about 50 to 90% of cation absorbing capacity of the soils to hold on to nutrient cations such as potassium, calcium, magnesium that may otherwise be lost by leaching. Its acid and base functional groups help with the pH buffering capacity of soils, and also it has a high water holding capacity, which helps with soil water retention. So we have looked at some of these important soil health parameters, and especially how they interact amongst each other and affect each other. The biological components affecting the aggregation in turn is affected also by organic matter. All of these are interconnected with each other. The importance of these soil properties towards soil health is known. However, it's very hard to establish a universal target value in absolute terms, since other outside factors such as climate, topography, hydrology, they may influence these soil properties. So the soil health assessment needs to include baseline or reference values in order to enable the identification of management effects on soil health. That is precisely the objective of Alberta Soil Health Benchmarking Project. This project will establish a soil health benchmark database for Alberta by obtaining a baseline of soil biological, physical, and chemical parameters associated with soil health across the province. The project will also monitor the effect of management practices on soil health parameters during the 3-year study period. One of the other objectives is also to improve the understanding of soil health parameters amongst the Alberta producers. A minimum of 220 sites will be sampled. These sites will include croplands, forages, pastures. There is no specific land use criteria for site selection. However, a record of site management history is needed. GPS coordinates will also be recorded for each of these sites. Farming Smarter is participating with other agricultural associations in this project, and each of the association will be responsible for sampling 20 locations Farmers can also contribute to the benchmark inventory samples if they wish. For each location, a representative area, about 1.5 acres, will be chosen depending on the uniformity of field conditions. Um, Aerial pictures can be used to choose these benchmark uh, areas. In these areas, compaction depths for the soil depths for a compaction at 300 psi will be measured at random points. If this compaction occurs for most samples at below 6 inches of soil depth, then the sample will be taken from 0 to 6 inches of depth. However, if the compaction is above 6 inches of soil depth, then the samples will be taken in two separate increments of 0 to 3 and 3 to 6 inches of soil depth. For these samples, these physical, chemical, and biological parameters will be analyzed either in the lab from the samples or in the field. Physical parameters include wet aggregation stability, compaction, bulk density, texture. In the biological parameters, there is active carbon and carbon nitrogen ratio, microbial respiration, and then the active and total populations of bacteria, fungi, and the nematode and protozoa functional groups. Chemical parameters, the general parameters that are tested in a soil testing lab, including organic matter, nitrogen, phosphorus, potassium, micronutrients, pH, electrical conductivity. All of these will be measured. All of this information will be entered into a database for the province of Alberta. Sampling has been done for 2019. It's ongoing for this year. It'll be repeated again the next year. And for these 3 years, the effect of management practices on these health parameters will be recorded. So this is my presentation for today. Thank you everybody for your attention, and I'm happy to answer if you have any questions.
Awesome, thanks Gurbir. Did anyone have any questions for him? Anything you're seeing in your soils at home?
Sorry, did you ask me, Jim?
No, I was just wondering if anybody is seeing anything out there in the soils that they're either managing or working themselves.
Okay.
Hey, Gurbir, I'm curious about how farmers can make the changes to improve soil quality. And when I ask the same question to the leader of this project, quite often they'll talk about these different cocktails of various types of plants and cover crops.
Mm-hmm.
I'm just, to me, that's not a really direct enough answer. It almost seems like it's sales orientated because I want to, sell a whole bunch of cover crop seeds and this and that. Is there any merit to this, growing a whole bunch of diversity of crops, or is our normal crop rotations okay at maintaining a decent, healthy soil?
Well, there has certainly been a move towards diversifying the crop rotations. I am not sure if a cocktail of crops, if we have a good enough data at this point, or if there are studies out there that I may not be aware of, that completely establish the usefulness of the cocktail of crops. There is definitely, it is definitely useful to have diversity in the crop rotations. That since single crop rotations or a given set sometimes can be limiting, especially to the microorganisms. So based on that, I do expect the cocktail cover crops to be useful. However, there are also other management practices. that are useful. I think one of the simple thumb rules is that if it benefits soil organic matter, it's generally good for soil health too, because soil organic matter is generally connected to pretty much everything else. So I think the move towards no tillage has been very good for the province and for Canada in general. I think it has increased organic matter levels and definitely improved soil health. Other other management practices like cover crops, no fallow, all of these are, I think, useful. Does that answer your question?
Yeah, I think that's good. Thank you.
Thanks.
All right, if there are no other questions for Gurbir, we will see you all at the next session, but feel free to reach out to us. And if you're a subscriber, you can see Gurbir's monthly Soil Savvy column in our subscriber newsletter.
The fungus Sclerotinia stem rot can be a threat to canola, each year causing yield losses across Western Canada. Get ahead of it with Quash fungicide. Assess your disease risk. If the conditions warrant a fungicide, then apply Quash. This Group 3 fungicide offers excellent Sclerotinia protection in a convenient liquid formulation. Apply at the 20 to 50% bloom stage. Quash absorbs quickly through the plant cuticle, spreading outward from the initial point of contact. Any more questions? We're here to help. Just ask a new farmer.